In animal cells, the lamin meshwork underlying the inner nuclear membrane helps keep nuclear pores evenly distributed throughout the nuclear envelope. When the nuclear lamina is disturbed, nuclear pore complexes cluster together and protein import into the nucleus is impaired. Fungi, however, don't produce a nuclear lamina, raising the question of how fungal nuclear pores are organised. Gero Steinberg from the University of Exeter in the United Kingdom became interested in this question when he noticed that nuclear pore complexes were highly mobile in the nuclear envelope of the pathogenic corn fungus Ustilago maydis.
At interface, we saw that the nuclear pores are moving around, and we just got interested in it. What is the mechanism behind this movement? And then, of course, why are they moving? So why do the fungal cells invest energy to move the whole nuclear pore complex, which is a huge assembly of proteins, in the nuclear envelope?
Steinberg et al. Saw that the nuclear pores in Ustilago maydis moved around randomly in the nuclear envelope with occasional rapid directed movements at speeds of about 1 micron per second. This directional motility could disperse nuclear pores away from each other and sometimes caused large deformations in the nuclear membrane. The speed and directionality of these movements suggested that the pores might be moving along microtubules.
We looked at the co-localization of microtubules and the nuclear pores, and we saw that the nuclear pores were moving along the microtubules. So we simply destroyed the microtubules by using fungicides, benomyl, and under these conditions the motility was almost completely abolished. And we also destroyed the actin cytoskeleton, and under these conditions actually the motility was not impaired. So these two approaches said it's microtubules.
Since the pore movements were inhibited by depleting cellular ATP levels, the researchers then investigated which motors were transporting the pores along microtubules. In Ustilago maydis sporidial cells, microtubule plus ends are oriented toward the cell poles, while the minus ends are located at the neck region between mother and daughter cells. Steinberg et al. Saw that the nuclear pores moved in both directions.
And so when a motility of a nuclear pore was directed towards the cell pole over the nucleus, we knew this must be a kinesin, and the motility towards the neck region must be dynein.
Sure enough, in Ustilago maydis cells lacking kinesin 1, plus end-directed movements were inhibited and the pores accumulated on the nuclear envelope near to the neck. In dynein mutants, on the other hand, minus end-directed movements were disrupted and the pores gathered near the cell pole. And in double mutants lacking both motor proteins, directed motility was completely blocked, and pore complexes clustered together in the nuclear envelope, indicating that bidirectional trafficking by dynein and kinesin 1 helps distribute nuclear pores evenly in the absence of a nuclear lamina.
We can see that the dynein passes the nucleus and then it seems to grab a nuclear pore, it pulls the nuclear pore towards minus ends, and then it continues moving, the nuclear pore is released and the nuclear pore returns to the nucleus. So it's just a transient interaction with the nuclear pore. Whether directly or indirectly, and what the adapters are, we don't know.
But what about other species of fungus, like Saccharomyces cerevisiae and Aspergillus nigellans? Do they also avoid clustering by moving their pores around in the nuclear envelope?
The motility also exists in these two fungi. So they also show directed movement at about 1 micron per second. It's in all three fungi the same velocity, but it looks like the molecular basis of this movement is different between the fungi. So budding yeast apparently is not using microtubules to move the nuclear pores. It's quite clear that the movement in the Saccharomyces cerevisiae is actin-based, and they also show the same consequence if we stop the movement, which is the clustering of the pores. So it seems like the process is there and is maintained, indicating it's an important process. However, the molecular machinery moving the nuclear pores might differ between the different fungi.
But why would fungi put so much energy into evenly distributing their pores? In lamin-deficient animal cells, nuclear pore clustering is accompanied by a reorganization of the cell's chromosomes.
it is known that chromosomes interact with nuclear pores, and we indeed found that the chromosomes move inside the nucleus. So we had to partially photobleach the nucleus and then see the chromosomes moving into the bleached area. And when we did this, we could also see that this correlated with the motility of nuclear pores. And then we simply blocked the motility of the pores, and when we did this, the chromosomes start to cluster at the periphery. So they are no longer moving, and the overall organization was quite remarkably and significantly changed. So this indicated that the motility of the pores organized the chromosomes inside the living interface nucleus.
Steinberg thought that disrupting pore motility and distribution might also inhibit transport between the nucleus and cytoplasm.
We tested import using GFP with the nuclear localization sequence, and we photobleached and looked for recovery of the GFP in the nucleus, which is an indicator of import. So in motor mutants or when the microtubules were gone, we found that the recovery and import was significantly impaired.
A photoactivatable GFP reporter took longer to move out of the nucleus when pore motility was blocked, suggesting that pore clustering also hinders nuclear export, perhaps because nuclear proteins and RNAs can only diffuse through chromatin-free channels in the nucleoplasm.
What I'm thinking now is that simply because the pores do not move, the pores cluster, this leads to chromosome clustering at the periphery, and that is affecting the diffusion channels. And this closing of the diffusion channels is actually the reason why the import-export doesn't work as good as it should. That's what I think in the moment. But there are alternatives.
The movement of fungal nuclear pores therefore seems to fulfill many of the functions performed by the nuclear lamina in animal cells, evenly distributing the pores around the nuclear envelope, organizing chromosomes, and optimizing nuclear import and export. The next question, says Steinberg, is to determine how the dynein and kinesin motors attach to the nuclear pores of Ustilago maydis.
And when we have the adapters, let's assume they are specific adapters between the pores and the motors, we can affect the transport specifically. So rather than taking motors out, which is a very crude thing to do because the motors do several other things, we specifically impair motility. If we have that, then we can go for more specific questions of the consequences. For example, is transcription affected? We don't know this yet.
In the meantime, you can learn more about how the motility of fungal nuclear pores organizes chromosomes and fosters nuclear cytoplasmic transport in the paper by Steinberg et al., published in the August 6, 2012 issue of the Journal of Cell Biology.
You